Okay, so today we will discuss amino acid and peptides. So the amino acid, as you can see here, that's basic structure of the amino acid, and this is general formula of all amino acid. Okay, and the predominant structure, ionic structure, the ionic form that predominate at the pH 7. Okay, and then you can see in the center here, that'll be the alpha carbon, and then always you will have a one amino group, one carboxyl group, and then you have this side chain, and the different amino acid basically they differ in the side chain, okay, because of the side chain that give us a different amino acid. All right, so this is the general structure, the general formula of an amino acid. And there are two stereoisomers of amino acid. So we have left and right, okay, L and D, right? L and D. So that's the stereochemistry of the amino acid. And with the exception of glycine, which we will discuss later, the structure of each individual amino acid, okay? All the protein-derived amino acids have at least one stereo center, okay? And so basically the center is alpha carbon and the chiral, the stereoisomer. And the vast majority of amino acids have the L configuration at the alpha carbon, and the protein is usually D, okay? So that's basically the general property for the amino acid. And this is the table just to show the common amino acid found in the living organism. The 20 amino acids found in the living organism, from alanine all the way to valine. And this is 3-letter abbreviation, 1-letter abbreviation. So you should know this, the connection of these 3-letter, 1-letter, right? Refer to the full name of the amino acid, okay? And the stereoisomer of the amino acid, and so the glycine is the only common amino acid that is not chiral. So that's because if we go to this slide, the glycine, oh, okay, here's glycine, all right? So we have amino group, carboxyl group, and this supposedly to be the side chain, all right? But this one is the simplest amino acid, it's only hydrogen ion, all right? Therefore, that's why the glycine lies not chiral, okay? Because you don't have the imaging, stereo imaging for the glycine. And so the majority of the amino acid that from the living organism isolated from the protein is L form, right? Yes, Diana.
Hi, I'm sorry. I just want to clarify. So glycine, you said that because they have a hydrogen instead of the side chain.
Side chain, yeah. That's why they don't have this stereo imaging right here. Look at this glycine, it's the simplest amino acid. All right. And
thank you.
You're welcome. And then this is how they identify these left and right, okay? So that's the based on this alpha carbon of the amino acid. So you have L form, D form. And then for the amino acid, we have 4 different classes, all right? Normally we say it's 4 classes. We have these polar, non-polar, negative charge, and positive charged amino acids. So that's 4 different classes of the amino acid. Okay. And the first one is a non-polar amino acid. non-polar amino acid. That means the side chain is non-polar. So alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan, and methionine. So these are the non-polar amino acid. Their side chain are non-polar, and then they prefer contact with one another over contact with water, and they are hydrophobic. And later on, when we discuss the tertiary structure of the protein, then now I will show you this. Because they are hydrophobic, then they will have the van der Waals interaction, right, to stabilize the tertiary structure, to contribute the tertiary structure. That's a hydrophobic amino acid. And the R group of protein is unique. It is actually bounded to the alpha amino group, forming a secondary amine. So that'll be proline. Let me see, where's the proline? So you can see the proline here. So this amino group, they form this structure here, right? That's very special, this proline. So that's the, it's unique because they form the secondary amine, right here, so the proline. And the phenylalanine has the hydrocarbon aromatic ring. That's a phenylalanine. See, you have this aromatic ring, and then tryptophan also has the ring. Okay, these two are special when we use the when we try to determine the protein concentration. And if we use the UV, and this can contribute UV 205, and then can help to determine the protein concentration, okay, the phenylalanine and tryptophan. And the methionine has a sulfur atom in the side chain, and tryptophan has an indole ring. And the methionine, the sulfur, is very special. The methionine, it's one of the only two that has a sulfur in their structure, right? So this is sulfur. The other one is cysteine. Cysteine can contribute to the disulfur bond that can stabilize the protein structure. So these are the non-polar hydrophobic amino acid. And then the second group is a neutral polar side chain, right? Polar neutral amino acid. They have the R group that have a high affinity for water, but are not ironing at the pH 7. Okay, and so basically, most of these amino acids associate with one another by the hydrogen bonding. And cysteine molecule form a disulfur bond with one another. All right, as I just mentioned in the previous slide, the only two amino acids that contain the sulfur now be methionine and the cysteine. And cysteine, they can form the disulfur bond, right, to stabilize the tertiary structure. And the serine, threonine, they have a polar hydroxy group, and tyrosine hydroxy group bound to the aromatic hydrocarbon group. And that's tyrosine here, all right, tyrosine. So for those that carry the aromatic group, now we'll be able to contribute to the UV detection, and for us to help us to determine the protein concentration. And the cysteine side chain contain the CO group. Okay, cysteine, this is CO group. All right, so they can form a disulfur bond with another cysteine. Right, and then glycine, asparagine and glutamine. So these two, they have this amide bond in side chain. Okay, this amide bond in the side chain here. Okay, so these are polar, uncharged, right? Polar, uncharged, polar, uncharged. Okay, and then the next group will be the acidic side chain, and they both basically have the carboxyl group in the side chain, so they can lose their proton and forming the carboxylate ion, and their amino acids are negative charged at the neutral pH, right? Negative charge. So it's here, they will lose the proton, so it will be negative charge. That's acidic, right? Because they are donate, they are the proton donor, So they are acidic amino acid, all right? So that's the negative charged amino acid. And then for the positive charged, now will be the basic side chain, histidine, lysine, arginine, all right? So basically the side chain are positive charge at pH 7, okay? And so these are the three basic positive charged amino acid, lysine, arginine, and the histidine, all right? So these are the three positive charged amino acid, and especially the lysine and arginine are very important in the living organism because they can help, they contribute the stabilization of the nucleosome, because the histone tail, they have a lot of lysine arginine residue, and because these are positive charge, so they can interact with DNA, so they can stabilize the nucleosome structure. Okay, so that's why these two are important in the living organism. So for the basicity, the amino group, okay, the average value of pK for an amino group is at 9.47, right? And the side chain arginine is considered a stronger base than the aliphatic amine, and the side chain imidazole group of histidine is a heterocyclic aromatic amine. So that's the basicity of those amino acids. So the summary of these amino acids. So for 19 out of 20, the alpha amino group is primary. For proline, it's a secondary, right? For proline, remember this structure in the proline here, all right? If only this amine group here, so all the other 19, they are primary except the proline, okay? And with the exception of glycine, the alpha carbon of each is a stereo center, right? The glycine is not a stereo center. And the isoleucine and threonine contain a second stereo center. So that's basically the summary of the amino acid. And these are just some uncommon amino acid. So for example, the collagen, so you have hydroxyl lysine and hydroxyl proline. So this hydroxyl proline, all right, you can see the comparison between the proline and hydroxyl proline, okay, especially in this location, you have the hydroxyl group, and then the hydroxyl lysine, you see this in this location, you have the hydroxyl group, all right, so these are basically, you can find these in the collagen, all right, in the connective tissue. All right, so this is uncommon amino acid. And then the ionization of the amino acid. The ionization of the amino acid, just like yesterday we discussed. So the one will contribute the donor, that will be acid, right? So HA here can lose the proton, all right? So it's an acid, and then there's a conjugate base, right? and then this one contribute the proton and go it this way. So this is acid, this is a conjugate base. Okay, and for amino acid without a charged group on side chain exists in neutral solution as a neutral ion with no net charge, right? So you will see this carboxyl group, amino group like this. Okay. So this is the structure that when it's in neutron ion isoelectric point, right? So it's a neutral, all right? So you have this amino group here, you have positive charge, and carboxyl group, you have negative charge here, all right? So so basically the total will be neutral at 0. And then if you go this direction, right? So you accept one proton, all right? So the carboxyl group accept one proton, then it become positive charge, and the pK is very low, right? It's acidic pKa, and then you become positive charge, plus one net charge. If you lose one proton, right, from the neutral, the isoelectric point, if you lose one proton, okay, lose one proton, that'll be here. The amino group lose one proton. All right, that's a pK2, and this is normally basic. All right, so the neutral and then cation form and anion form, right? And anion form is negative, cation form is positive. In the middle, that's the neutral ion. And this is the example of the histidine, right? The histidine. That histidine contain the titrable side chain, all right? So this isoelectric neutron ion, all right? Between it's here, there's 0 net charge, all right? And then 0 net charge, you can see this is a amino group positive, carboxyl group negative, and then if you lose a proton, all right? If you lose one proton, become the -1 charge. -1 charge here, right? You lose a proton, and so you have -1. If you obtain one proton, all right, and this proton, okay, this proton doesn't go to the carboxyl group; they go to the side chain here, all right. So here will be positive, and then you obtain another proton, then now go to carboxyl group here. Okay. So that's because the histidine has the titraceable side chain. That's why you have these different formula here, right? And the neutral ion, so basically the two charge group, so the basic amino group and the carboxylic acid, and the 2N lead to the internal proton transfer. So that's called neutral ion. Okay. And then when you change the pH, then you can affect the net charge of the neutral ion. The chemistry of amino acid is complicated because the amino group is base and the carboxyl is acidic, because these contribute, this is a proton donor and this is a proton acceptor, all right? Proton acceptor, so it's base and it's acid. And the pH at which there is no net charge on the neutral ion, we call it isoelectric point, all right? So the isoelectric point, all right, isoelectric point, the net charge is 0. And this is just to show you the titration of the amino acid, that the titration here for these amino acid that's no titraceable side chain, it's simple. And the example here is a histidine with the titraceable side chain. It's a little bit complicated, all right? So this is just to show you this the titration of an amino acid. It represents the reaction of each functional group with the hydroxide ion. Okay. So isoelectric point, it's here, and you can see this amino group, all right, amino group, the carboxyl group, all right. So you have one positive charge and one negative charge, total is neutral, 0, okay. And then if you increase the pH, when you increase the pH, so you lose the proton, all right? You lose a proton. So from here, 0, and here will be a negative charge. And in the middle, that's the pK2 in the middle, then you have the 1-to-1 ratio, all right? 1-to-1 ratio of this and this, okay? And then if you are above the pH, above this point, then the predominant will be this one, all right? And then if it's below this pK2, then the predominant will be this one, right? The neutral one, okay? In the middle, in the midpoint, that'll be 1-to-1 ratio. And then for the from the ice, yes, Diana.
Can you just go over the net charge of the pK2 that equals 9.69 and how we could see that?
Well, here, right?
Yeah.
At this point, you have a 1-to-1 ratio of this neutral amino acid and -1, all right? Because that's if you increase the pH, then this amount will reduce, all right? And this amount, this one will increase. And when you reach the midpoint, you have 1-to-1 ratio. If you pass over this point, then the dominant will be this one, -1 charge, all right? And it's the same between this pi and the pK1, right? The pK1, that's the midpoint between this neutral and the positive charged amino acid. At the pK1, the midpoint, the ratio of the neutral amino acid and the +1 charge amino acid is 1-to-1 at this point, okay? If below this pH, then the dominant would be the +1.
Okay, now I see. Thank you.
Okay, so this is the titration of amino acid. And then this is titration of a histidine with a sodium hydroxide. So the histidine has a titraceable side chain, all right, as I show you here. So the titraceable side chain is here, all right. And then the pH 7, you have this net charge here, the net charge, right? And then you have this -1, right? You have a +1, +2. And then if we go back to this here, again, if you go from the pi, the isoelectric point, the net charge is 0, right? +1, -1, so it's 0. And then if you increase the pH, so that means you add more hydroxy group, all right? So you increase the pH, right, to the 9.2, that's pK3. So at the 9.2, the pK3, 9.2, at this point, the amount of the neutral amino acid and the -1 amino acid will be equal. That's 1-to-1 ratio. And then if you continue to titrate the histidine with the sodium hydroxide, if it passes the 9.2, let's say it's a pH 10, all right? When it reach to the pH 10 here, then the majority of the amino acid will be -1. All right, -1. Okay, if the pH below the 9.2, then the majority will be the neutral. And then between this, yes, Diana.
I'm sorry, I don't know why that happened.
Oh, it's okay. Oh, Hadel.
the exam or the quiz, right?
Um,
How would it, like, what would the question, like, it would tell us to draw, like,
Oh, very good, Heidi. So the question is, I will ask you at the pH 7. At the pH 7, what would be the dominant for the specific amino acid? Or the pH 4, then what would be the dominant amino acid form? Okay, so that will be the kind of a question. All right. So this is just an example to show you, as you increase the amount of hydroxy group during the titration, you add more sodium hydroxide, right? And then you increase the pH. All right. So we can go from the pH is from pH 1. All right. So from pH 1, between pH 1 and pH 2. All right. And there's a pK1 1.82. So at this point, the at this point 1.82, the amount of the plus 2, see, 2 positive, the amount of 2 plus 2, all right, versus the plus 1. This cancel out, so you have plus 1. So plus 2 plus 1. If it's a pH 2, if it's pH 2, then the plus 1 will be dominant, all right, because at the pK1 1.82, the amount of the positive 1 and the positive 2 will be equal at the pK1. All right. And then in this range between pK1 and pK2, all right, so that would be mainly positive 1 form. All right. And then between pK2 and pK3, all right, that would be the dominant is pi here. That would be a neutral, okay, the neutral. Then now depend on at which point. So if it's at pK2, then the amount of the neutral and the positive 1 will be equal. All right. So if it's below 6.0, then the positive 1 will be dominant. Above 6.0, then the neutral will be dominant. Okay, that's how you see this, Hadil. Yes.
be able to calculate that from just you giving us, telling us, oh, at
This
pH,
number,
which one would be,
in the next slide, Hadil, for this number, pK1, pK2, pK3, now be in this table. All right, so this is the pKa value already determined. So just based on this number, then you can predict what's the dominant form of the amino acid at a certain pH. All right. So that histidine, we have these three pKa values. So this carboxyl group here is 1.82. All right. Histidine 1.82, 6.0, 9.17. So that will be 182, 6.0, 9.2. So you know 1, 2, 3. All right. And then the pi here, that's a 0, and then you have -1, +1. This is +1 and +2, right? +2, +1, and neutral and -1. And then if you are asking, okay, pH 5, what would be the dominant form? The pH 5 will be here, so the dominant form will be this one. Okay, and this is the pKa value. All right, so this column, that's for carboxyl group, and this column for the amino group, and this column is for the titraceable side chain. All right. So basically the value, yes, Julian.
Sorry, Professor. So just to get an idea, so basically you would have a chart like this, and we would.
Yes. You will have a chart like this, and then you will just to think what would be the majority form of the amino acid at a certain pH. based on this table, this the pKa value table. Adele.
There's different values at different areas. So the pKa at for COOH, it will be dominant at 1.82, and for the 3
Yes,
plus, it will be
below,
dominant at like the 9.2. Is that what you're saying?
okay. So pK1 is 1.82, right? So below 1.82, the dominant form would be this one, positive 2. And then if it's above 1.82, then the dominant form will be this one, positive 1. At this point, at least 1.82 point, at this exact pH, then the amount of this plus 1 and the amount of plus 2 will be equal. All right, Hadil? Mona.
So how will we know the pH value? Because I see the pH.
No, the pH, yeah, the pH will be given. So that if there's a question, then I would say, okay, at the pH 2, then let's say at the pH 2, then we have amino acid glycine. What will be the dominant form? Do you see what I mean? And then if you look at by looking at this, so the
Yes.
pH 2 would be below this value. All right, so the dominant form would be positive 1.
Oh,
All right,
got it.
do you understand? If we go to this simple one here, that's this one. This is what's this one? What's the amino acid? It doesn't say, but okay, 2.34. Let's say that's this one, glycine. All right,
Yes.
2.34 glycine, and 9.69. This is glycine. All right.
Yes.
So if it's a pH 2, so pH 2 that below this pK1, all right, below this 2.34, it's here. So the dominant form will be plus 1. All right. And then if you have the pH 2.34, which will be exactly the same as pK1, so at this point, the amount of positive 1 and the neutral will be equal.
Equal. Got it.
Yeah. If the pH, if we give you pH 4, which will be here, the pH 4 will be at this point here. And this point, then the dominant form. So what will be the dominant form? The dominant form will be the neutral.
Neutral. Yeah, got it.
Yeah.
Okay.
Okay. Good. Yes, Mona, again.
I didn't do it. Sorry.
Oh, okay, no problem. Okay, so that's the how you predict the the majority form of the amino acid, all right, during the titration. Okay, so that's the titration of amino acid.
Yeah.
And the value of a carboxyl group are fairly low, so you can see here it's acidic, all right? And the value of amino group are much higher. It's all in here in basic pH. And the value of side chain group, including the side chain carboxyl and amino group, depend on the group's chemical nature. All right. We will have this summary later. But this slide is too complicated. You can skip, all right? And then isoelectric point, isoelectric pH, all right, the PI. So that would be the pH at which the majority of molecule of a compound in solution have no net charge. So the PI would be half of the pK1 plus pK2. Okay, so for the glycine, let's say for glycine here, for glycine, the PI, all right, the PI would be 2.34 plus 9.6 divided by 2. All right, so that's what they did here. This is 2.35 and 9.78, and then divide by 2, 606. So this is the PI, all right, for glycine. So for to determine the PI for all these amino acids until protein here, all right, that's very simple. you just add the value, the first value from the first column and the value from the second column and divided by 2. That'll be pi. All right. And then from here to arginine, all right, these 7 amino acids will be a little bit complicated that we would explain here. So first of all, for those simple amino acids, only 2 pK will be simple. Pi equal to the total, the sum of pK1 and the pK2 divided by 2. All right, no problem with this. Okay, now for the for these those with the traceable side chain. Okay, so first of all, these two, ASP and GLU glutamine, right? So the average, so the side chain is acidic, all right? The average, the side chain pK with the side chain pK and the carboxyl pKa. That will give you the pi. All right, so for these two, all right, so you have a carboxyl. So that'll be these two, all right, 2.09, 2.19, all right? And then this side chain here. All right, so you take the average of these. Yes, harder. Harder. Hey, Dale.
Yeah, so why do we take the one with RH or RH plus? Why do we take the average of that?
Because that depends on the special function, the special property of the side chain, so that it depends on the how they obtain the proton, lose the proton. So then, for example, for the histidine, arginine, lysine, all right, histidine, arginine, lysine. So for these three, all right, the side chain that you will take the pKa of the side chain with the amino group. All right, so now be this side chain and the amino group, right? Because these three, you have positive 2, positive 1, 0, and negative 1. And for these two, you have negative 2, negative 1, 0, and positive 1. All right, so these two are completely different. So
it depends on the side chain, right? Like what side chain.
Right.
Okay.
These three will give you +2, +1, 0, and -1. All right, for these three, and for these two will give you +1, 0, -1, -2. Okay, and then for our ionizable group, the tyrosine cysteine, these two. All right, for these two, you determine which is the middle pKa and average with the carboxyl pKa, right? As a tyrosine and cysteine. So basically that'll be a 911 plus 2.2, and then take the average of these two, all right? So that's for this tyrosine cysteine. So that's the 3 different calculation for these 7 different amino acids that carry the titraceable side chain. Okay, that's how you determine the PI, the isoelectric point. And then for the peptides, all right, so that's individual amino acid. And then for the peptide, find the approximate pH where the net charge will be 0, and then average the closest pKa value on either side of this value. Okay, and then so if the side chain is acidic, so you average the pK with the carboxyl groups. Now we just explained, and these three is pK, side chain pK, and amino group. And then ionizable tyrosine, cysteine, you determine the middle pK, average with the carboxyl group, right? So the glycine, so you have very simple, you have only 2 values. You take the average. And then for the ASP here, so that's this one. So pK, the side chain pK is carboxyl group, okay, carboxyl group and side chain pK. So the PI will be this, all right. And cysteine, all right, the side chain with carboxyl group. And the lysine will be the amino group here with the side chain. So this is a PI. Okay, so this is how you determine the isoelectric point. Okay, and then for peptide, all right, for the peptide. Now, if we have 3 peptides, normally when you write from the left to right, and then we know this one will be the N terminal. In here will be the C terminal. Okay, N terminal to C terminal. Okay, N terminal to C terminal. And then to calculate the PI for this peptide, for these 3 amino acids that give you the peptide, right? And the peptide, so basically you have amino group, carboxyl group, and then the middle one, you have to see if the middle one carry the side chain or not, all right? And unfortunately, this one carry the side chain, so glycine, the amino group, the glycine, all right? Amino group here. And then on the right side, the C terminal, carboxyl group, the glutamine here. So you have 2.1, 9.78, right? And now you have this side chain for this one and this one. Okay, and then you should put the side chain pKa for the side chain here, and then you add everything together, and then you see this number, all right? And then you find the middle number here, all right? And then you take the average. That will be the PI. Okay, so for these 3 amino acid peptide, all right? And then always the left side will be N terminal, the right side will be C terminal. So the N terminal, so you look at amino group pKa for amino group for glycine. So you put it here, 9.78, and glutamine, so it's a C terminal. So you have carboxyl group pKa 2.10. So you put these 2 numbers here. And then the next, you look at the side chain. So now the side chain for aspartame is 3.86, glutamine 4.07, and then from these different pKa, then you find the middle 2 here, right? From 210, 386, 407, 978, these 2 are in the middle. So you take the average of these 2, that gives you the PI. All right, any question? Any question? No, very good. Good. And then if we have 4 amino acids, all right, in this peptide, again, the on the left side here is amino, the N terminal, C terminal. So cysteine, all right, cysteine, you put the amino Ka for the amino group, and then arginine, all right, there's a carboxyl group here. This, all right, so you put these 2 numbers here, okay, and then you just examine each of individual one to see if they have the side chain. So since they all have the side chain, so you put the side chain of pKa here, all right? And then you pick these 2 in the middle, all right? So 2.01, 407, all right, and then 8, and 10.25, 1053, 1248. So these 2 numbers are in the middle. So you take these 2 numbers and average these 2 numbers. That give you the PI for this peptide. Okay, any question?
What 2 numbers?
The 2 numbers in the middle.
Okay.
So first of all,
Why
you
those
check.
two numbers? Like, why exactly those two numbers? Why didn't we take the pKa5? That's closer. I mean, like, that's at least like close to the 1025. Why didn't we take 1053 with it?
No, you don't take 1053 because you have to take the pK number in the middle, and the 1053, although it's close to 1025, but it's not in the middle. I mean, to calculate the pi for the peptide, you have to follow this rule. Yeah, because these cysteine, this pH, the pK8 here, pK8 here, cysteine here, that can contribute to the the ionization greatly. So that's why they developed this formula to calculate the pi. And
What
you
final
can
numbers are we looking at? Like we're looking in between 10.25 and what to determine the pK?
no. Okay, so for this six number, if you make the arrangement from the lowest number to the highest number, so you will see the first number 1, all right, 2, and this is number 3, and number 4, number 5, number 6. So that's why you take the number 3 and number 4 number that's in the middle. These two numbers, number 3 and number 4 in the middle, and then you have to average.
Is that because there's 4 peptides?
4 amino acid, and then
Yeah, 4 amino acids.
per amino acid, like a 5 median, yes. And then first of all, you have to get the amino pK for amino group here, and pK for the carboxyl group here. Then you look at the titraceable side chain. All right. So since each of these contribute, they all have the titraceable side chain. So you have to put the pK for those titraceable side chain here. So you have 6 number. For these 6 number, then you find the middle number 3 and number 4 number.
Thank you.
Okay, good. So that's the PI calculation. Okay. And then the peptide bond. The peptide bond, so basically the individual amino acid can be linked by forming the covalent bond, all right? And they just like the nucleic acid, okay, 5 prime to 3 prime. For the amino acid, we have this amino group to the C terminal, the N terminal to C terminal. N terminal always put on the left side, C terminal on the right side. So it's just like a DNA 5 prime to 3 prime. You always write the 5 prime from left to right, all right? And then they can, these 2 amino acids, they can form the peptide bond by dehydration, right? You can dehydrate 1 molecule water, all right, and then you connect these. You have formed the amine bond between the alpha carboxyl group of 1 amino acid and the alpha amino group of another amino acid, all right, and then that's a CONH, all right, CONH bond, the peptide bond, all right. This is from the first amino acid, is a carboxyl group, and the NH, that's from the amino group of the second amino acid. So they form these peptide bonds here, okay, through the dehydration, remove 1 water molecule and form the peptide bond, and then you can continue to do these synthesis through the dehydration, and then you can have many amino acids to add it to the C terminal end, okay? And so this is direction peptide chain, all right, from left to right, and N terminal and C terminal, and this is a peptide bond. So you have this CONH, all right, and then CONH, so you can continue to synthesize the peptide. And this is also the way in the living organism when they perform the translation, all right. So that's the direction also to synthesize the peptide chain, okay. All right, so that's the end of the chapter 3. Do you have any question?